How does the MHC exogenous pathway work in immunity? Imagine your immune system as a busy security team constantly scanning for intruders. One of its key methods involves showing pieces of outside invaders to alert the rest of the immune system. That's where the MHC exogenous pathway comes into play. It's a process used by special immune cells, called antigen-presenting cells, to display fragments of proteins they've taken from outside the cell. These fragments, or peptides, come from bacteria, parasites, or other foreign substances that the cell has engulfed. First, the antigen-presenting cell engulfs extracellular proteins through a process called endocytosis. Inside the cell, these proteins are broken down into smaller pieces called peptides by enzymes that work best in acidic environments. Next, the cell prepares its MHC class Roman II molecules, which are made in the endoplasmic reticulum. These molecules have their peptide binding sites blocked by a protein called the invariant chain to prevent them from binding to random peptides too early. The MHC class Roman II molecules, still attached to the invariant chain, are then transported in vesicles from the endoplasmic reticulum to fuse with the endosomes containing the peptides. Inside these vesicles, the invariant chain is gradually degraded, leaving a small fragment called CLIP in the peptide binding groove. A molecule named HLA-DM helps remove CLIP and assists in loading the MHC class Roman II molecules with the peptides derived from the outside proteins. Once the MHC class Roman II molecules are loaded with the peptides, they are transported to the cell surface. Here, they display the foreign fragments to helper T cells, specifically CD for plus T cells. This recognition is vital because it triggers the helper T cells to activate other parts of the immune system, like B cells for antibody production or macrophages for engulfing pathogens. This pathway is especially important for defending against bacteria and parasites that live outside cells. It mainly occurs in professional antigen-presenting cells such as dendritic cells, macrophages, and B cells. Sometimes, these cells can also perform cross-presentation, where they present external antigens on MHC class I molecules to activate cytotoxic T cells, which target infected or abnormal cells. In essence, the MHC exogenous pathway helps the immune system keep an eye on the environment outside cells. It processes external proteins, loads their fragments onto MHC class Roman II molecules, and displays them on the cell surface. This process alerts helper T cells and sets off a chain of immune responses to protect the body from invading pathogens.